then we can get your
Sure, yeah,
slide.
of course. Yeah, go for it.
So Dr. Samra Turajlic is going to talk to us about research-based career options today, and she completed her undergraduate studies in Oxford and clinical training at UCL Medical School. Her PhD was obtained from the Institute of Cancer Research in 2013 in the field of melanoma genetics and targeted therapy resistance, and she's a consultant medical oncologist on the skin and neurology units at the Royal Marsden. Dr. Turajlic became an independent group leader at the Francis Crick Institute in 2019 and divides her time between the clinic and laboratory. She is also the chief investigator of translational studies in melanoma and kidney cancer, in particular the TRACER-X study, and her research goal is to develop an evolutionary understanding of cancer for patient benefit. She's a member of multiple groups, including the NCRI Bladder and Renal Cancer Clinical Studies Groups, an ESMO faculty member for genitourinary cancers, and a scientific committee member for ESMO Molecular Analysis for Personalised Treatment. She's also part of the Kidney Cancer Association and the EORTC NCI AACR Symposia. So without further ado, I'd like to introduce Dr. Turajlic to talk to us today, and if anyone does have any questions, we'll ask these at the end. Thank you.
Thanks, Abby, for the kind introduction, and thank you, Johnny, and everyone for the invitation. I hope that this session will be useful to you all. Now, I'd like to keep it as informal as possible. So if you do have questions as we go through, I have about a dozen slides, not more. I mean, I hope that we can just focus on the discussion mostly, but feel free to use the chat function as well if you'd rather not come on and ask your question that way. So let me just share my screen. Let's see if I can go to slide. Can you see that okay? Okay, so as Abby says, I wear two hats. I work as an oncologist at the Royal Marsden, and then I lead the research group at the Francis Crick Institute. And Johnny gave me quite a broad remit, which I thought was quite challenging to think about all the possible research careers in oncology. I will mostly really speak from my own perspective, which is that of a clinician scientist. But I guess for all of us, you know, when we're considering these decisions, what's very important is the signposting, and then also what you have in front of you in terms of senior colleagues that really illustrate what a career might look like. So when I thought about this from the perspective of sort of clinical research or service delivery, it really is very apparent what those pathways might look like. So if you're focusing very much on a clinical career, you'll be involved in service delivery, but then also increasingly as you become more senior in education and management, and your trajectory might lead to being a clinical or medical director, and then in the context of education, perhaps director of medical education or a training program. If you have protected clinical research time and you're involved in either academically initiated studies or commercially driven registration studies, you might develop very close network with pharma. You'd be a KOL in your lead, in your field. You might become a research lead in your cancer group, director of R&D, and so on and so forth. But when it comes to academic careers, there is, in my experience, a lot less kind of visibility of what really happens between your PhD and then a potential subsequent trajectory. Although I realize with the more structured program now, hopefully that is a little bit more apparent to you who are entering the field. And that's partly to do with the fact that there are so few clinician scientists generally, and probably each one of them has had a fairly unique trajectory to the position that they're in today. But then also because a career in academia can also be very broad in terms of its subject. So if you're working in basic research, this could be quite disconnected to what your clinical interest is. And certainly if you look at last year's Nobel laureates in physiology and medicine, it was impressive to see that they were all clinician scientists, but they were all driven by fairly basic questions and weren't necessarily working on the same problem in their clinical practice. And then, of course, if you're working in drug discovery or closer to translational research, or perhaps biomarker-driven research, your network might be much closer to pharma than other basic researchers. I hope to shed a little bit of light on a potential path from research earlier on in your training to becoming a research group leader, and how that might integrate with your clinical practice. So this is my own path to this stage, and admittedly, this was all done in the old system. So I'm really hoping that things run much more smoothly for you now. There isn't so much reliance on kind of own initiative to find out about these opportunities. But just to say that I really didn't get into research until about halfway through my training in medical oncology. And in fact, I hadn't been set on a career in academic medicine at all. I was really enjoying my clinical training. And it was only when I started in medical oncology, and in fact found myself treating melanoma, which I know this will seem unfathomable today, given how the treatment landscape for melanoma has moved, and what a poster child it's become for your potential cure in solid tumor oncology. But all I was doing during that time is essentially acting as a palliative care doctor and giving the carbazine and temozolomide to patients with metastatic melanoma. And I was very lucky that at that point there was an opportunity in the lab of Richard Murray, the Institute of Cancer Research, for a clinical research fellow to undertake a PhD. Now, what was exciting was that this is the lab that had worked out the crystal structure of BRAF a few years previously, and that was in the back of the discovery that 50% of melanomas harbored the BRAF V600D mutation, and how that amplified the kinase activity. The crystal structure then allowed for the development of the drug, and I think this was, I was extremely fortunate that this was a time when it was very clear how doing research could interact with clinical practice, because the preclinical findings were so close to what was happening in the clinic. So I joined this lab that was focusing on BRAF signaling at a time where next door at the Royal Marsden, my colleagues were running phase 3 trials of BRAF inhibitors in BRAF mutant melanoma. And just to give you an example of that proximity, you will, some of you might remember, or others may have read about the fact that the patients that were treated with BRAF inhibitors as monotherapy originally started developing these skin lesions, some of which were cancerous. And it had become apparent that these skin lesions were coming about as a result of BRAF inhibition. So around that time, the lab that I was in had discovered something called the paradoxical activation of the MAP kinase pathway by BRAF inhibitors in the context of NRAS mutations. And it quickly had become apparent that what was happening in the patients is that their keratinocytes, which are the most abundant cells in the skin, were harboring these RAS mutations. And once you applied the BRAF inhibitor, you would cause this paradoxical activation of the pathway, and they would turn into tumors. And then in the context of what I was working on, which were the rare melanoma subtypes, and really I very much joined a fundamental science lab. They were focused on cellular signaling, and my project was very much around in vitro and in vivo models of rare melanoma subtypes, including KIT mutant melanoma. So I had a very broad exposure to basic science principles, but as well as methods and training. And only in the latter part of my PhD moved into next generation sequencing, which at that time was a completely novel development in cancer genomics. And again, it's amazing to see today that it's something that really is at our fingertips all the time. But what really interested me was something that I picked up in the clinic was that the patients who were receiving BRAF inhibitors, and again, this today seems very intuitive, that of course they were going to develop resistance because the selective pressure of targeted therapy would drive that resistance. And I was then able to develop PDX-derived tumor models of BRAF inhibitor-resistant melanoma, and therefore we had the insight, one of the first insights into mechanism of resistance to targeted therapy. And what was also obvious was that this was all being fueled by intratumor heterogeneity, which was a consequence of cancer evolution, and something that at that point we began to appreciate was pervasive in all solid tumors. And although I very much had enjoyed melanoma biology, and the way that I'd engrossed myself in that for 4 years, was really very compelling for me. The opportunity to study cancer evolution in the context of another cancer model, which was kidney cancer, was very attractive to me, and therefore I decided to develop my Cancer Research UK Clinician Scientist Fellowship around the subject of cancer evolution. And there are many reasons why kidney cancer was a more compelling model in this setting that I'm happy to discuss later on, but it's not really the point of this. What I really wanted to emphasize is that I had a very broad research training, and really my focus between 2009 2013 was to get the most out of my time in the lab, and really still undecided at that stage whether I would carry on. But I was bitten by the science bug, and when I completed my PhD, I just thought that I couldn't possibly go back to pure clinical work and never do science again. I just could not imagine that. So it was a choice that was really informed by passion rather than any kind of strategy. And a natural next step here would have been to have applied for a clinical lectureship. But I was in a slightly peculiar situation in that I had become very embedded with a melanoma unit at the Royal Marsden. And I think I'll come back to this later on. I think really finding a clinical unit that's going to be your home while you do research is extremely important, and that you might build on those foundations as you progress through your academic career. So I very much wanted to remain with them at the Marsden, but I was keen for this next step, which was a semi-independent step through a clinician scientist fellowship, to go to the London Research Institute, which has since become the Francis Crick Institute, and joined the lab of Charlie Swanton, who was working on cancer evolution, and was at the time was really the lab to integrate oneself in to understand the subject. So I had to, by necessity, skip the CL stage because I could not do a CL at the LRI and do my clinical training at the Marsden. So that forced me to develop my own proposal for the following 5 years of what I would work on. And I think in retrospect that was very helpful because it really clarified my vision about the research that I wanted to do. And it also gave me an opportunity to essentially become independent very early on. So what the CIOK Clinician Scientist Fellowship afforded me was not just my own support, but also ability to employ a postdoc and also quite generous research funding. And then you can really leverage that to establish other collaborations and attract other external funding. And then in the meanwhile, I was appointed at the Royal Marsden as a consultant oncologist, and in fact my primary employment is at the Royal Marsden, and I'm seconded to the Francis Crick to do my research. When the 5-year fellowship had finished at COK, I applied for the Advanced Clinician Scientist Fellowship, which again funds part of my group, provides my personal funding in my program. And then in parallel to that, I had also applied to become an independent group leader at the Francis Crick Institute. So both those things happened last year. And then earlier this year, I become a reader at the Institute of Cancer Research, which is Marsden's sort of, sister research institute. So this is what it looks like now. Our group at the Crick is called the Cancer Dynamics Laboratory, and we have a very rich program and a very broad program for a relatively young research group, which again is a consequence of the fact that for the last 5 years I've been developing a program that was semi-independent. So by the time I moved into my own lab, I brought a group with me. And we work essentially in these two disease models that are informed by my clinical practice, so kidney cancer and melanoma. But we work on everything from trying to gain fundamental insights from functional experiments, and that's in preclinical models, to interrogating large genomic data sets, and then to biomarker discovery and validation. So you're really working at right at the other end with clinical trials and embedding translational endpoints into those. The team consists of laboratory research scientists, students, and postdocs, and a number of my students are clinical students. And we have been very fortunate in the last year or so to have attracted around £5 million of funding to work across this research program. Institutionally at the Crick, I've got multiple responsibilities, obviously to my own group, but also feeding into various faculty roles as well. And we collaborate very widely at the Crick and its university partners, but also with many colleagues at the Marsden, at the ICR, and externally we collaborate with a group at NIH, but then also with industry, especially through immuno-oncology networks. Then on the clinical side, I provide a clinical service for melanoma and kidney cancer, so in person, 1 whole day a week. And then I attend multidisciplinary team meetings for both these diseases. And then I'm intimately involved with our research strategy on our unit, but also our institutional research strategy via BRC and ICR. What I've tried to do is really develop this translational research platform on our unit that would then inform what we do in the lab. And really I try to integrate the two things as much as possible. These are some of the studies that we're running at the moment. But at the same time, when my colleagues who are more involved in clinical research start investigator-initiated trials, what we try and do is really embed biological endpoints into those. So here's a number of studies where I've worked very closely with clinical colleagues to make sure that we really glean maximum insights from patients that we enter into these studies. And that really requires a separate translational protocol to go alongside the investigational product. And then very occasionally I'll be the principal investigator on a clinical study. So for example, I'm the national chief investigator on a trial called THETIS, and this is a first in human study of clonal neoantigen reactive T cells in melanoma. And the reason for really getting involved with this and leading on it is the fact that at the heart of this program is understanding of cancer evolution with respect to immunogenicity of different mutations, something that's very close to my heart and my program. And therefore it was important for me to get involved in this, and I certainly enjoy being involved in the clinical side of the realization of this product. And then some of the other sort of external groups that we work with have come about through the Genomics England 100,000 Genome Project, especially in kidney cancer and melanoma. And there we work in the context of GSIPs, which I think some of you would have come across or been involved with. So why do it? And nowadays a lot of people ask me this either from the point of view of my own position or their position, probably the answer changes depending on how well things are going, and certainly science can be a real roller coaster ride. But for me, the thing that was a real deciding factor when I finished my PhD and thought about the next steps, what was really compelling was the idea that you can generate fundamental insights, and you can generate completely novel insights into cancer that they're inspired by your clinical questions and your clinical observations, and yet not let go of your passion for engaging cancer patients and delivering clinical service. Of course, there's an opportunity to translate your findings, and this is often much easier for practicing clinicians than it is for basic scientists. Firstly, because we have a sense of what's relevant in the clinic, but we also know we're much closer to the roadmap for translation, and the opportunities that are open to us are often not open to basic scientists. So it's an incredibly privileged situation to be in. And when it comes to, if you end up working in a particular cancer type, you can really engage and contribute both nationally and internationally to the strategy, really the fundamental strategy to addressing that disease, whether that might be from an epidemiological point of view or treatment point of view or a basic insight point of view. It also means that you have a lot of freedom. I mean, it's very I think it's extremely liberating to go into science. Of course, it comes with a huge amount of responsibility to meet your milestones and responsibilities, but you can respond to emergent problems. So just to give you an example, at the earlier in the year, in response to the issues faced by cancer patients in the context of the COVID pandemic, and especially some of the catastrophic stuff around changes to cancer therapies on the assumption that cancer patients were more susceptible to COVID, and what that will mean for cancer outcomes longer term. We repurposed some of our research program towards understanding fundamentally the immune response to SARS-CoV-2 in cancer patients, and understanding how cancer interventions can impact that. Now, you're really only able to do that if you're in charge of your own research program and your research team. And I was again very fortunate to work with a very enthusiastic group, my research group at the CRIC, and also very responsive colleagues on the clinical unit who over the years become very used to the sort of translational research framework, and we were able to enact something that was really very impressive at the time. And another example is that through treating melanoma with IO over the years, we've become acutely aware, and all of you will be, of the issue of immune-related adverse events, and the deep dissatisfaction with the way that we approach and understand the mechanisms of those. So inspired by that, we've initiated again a systematic, a large-scale study into understanding these phenomena of essentially autoimmunity or autoinflammation induction. And of course, what gets you out of bed in the morning is that you're able to satisfy your curiosity, and it can be a lot of fun. But as with all jobs, over time, there is quite a considerable burden of things that are not fun, which are more management and sort of team running based, rather than really engrossing yourself in the science. Which is why I always think back to my PhD as a sort of a challenging time, but really time to have your head in that space without having to worry about too much else. But what's really challenging in this is it's quite hard to actually belong fully to either of the communities, so the science community or the clinical community, and both might regard you, although it's not been my personal experience, but I think this is perfectly possible, with some suspicion or skepticism. So if you're in a very basic science community, they might view your approach to research perhaps not fundamental enough. And then from your clinical colleagues who are involved in clinical management of patients all the time, perhaps might not view you as a sort of an equal member of the clinical team. And the truth of it is that you probably can't do the best of either of the jobs. But so the strategy really should be how to combine them in a way where they're greater than the sum of two parts. And this is another question that sort of comes up when I talk to trainees. So I thought it would be good to share my thoughts with you and hear what you think as well. So when it comes to choosing your question or field, again, we're really handicapped as clinicians because we don't have this sort of exposure to research environment or the intellectual process, if you like, throughout our training. So if we compare ourselves to basic scientists or perhaps the MB PhD candidates, we really go down a route of learning clinical medicine, which is very, very different. And then having to kind of unlearn that in order to liberate ourselves intellectually to ask probing and often fundamental research questions. So it's important that your research question is authentic. It's very easy to be told, oh, there is this good opportunity, and this is what this PI is interested in, and you should just kind of slot yourself into their interest and into their vision. But actually having something that's at least in part informed by your own clinical observations is very important. Being aware of the emerging clinical problems and where the knowledge gaps are is important, but also horizon scanning. So, as an example, liquid biopsies are, very interesting and very hot, but there's so many people working on that. You need to think, well, okay, this is something that's going to be kind of a standard of care approach in 2, 3, 4 years. So, what can I contribute if I get involved in that? What will be the next question beyond that? And then when you identify something in your clinical observations, and for me in the context of kidney cancer, what really intrigued me was just how varied the clinical manifestations of kidney cancer are. So we have these patients that have curative surgery and they never relapse, and then we have patients who might have a solitary metastases 10, 15, 20 years after having the primary tumor removed. Then we have the patients with oligometastases, maybe 3 or 4 years after the primary tumor removal. And then we have this group of patients who just explode with disseminated disease. So why is that? And actually in our case, we were able to apply, very pleasingly, this kind of evolutionary framework to really rationalize that clinical behavior. And that one is, it's very satisfying, but two, it actually then gives some credibility to the idea that the knowledge that you've created can be taken back to kind of predict the trajectory of individual cancers. So how do we get ourselves to a place of comfort to choose the question or the field? Well, it's really important to know how science is done. And I know that now in the more structured pathway, there is more exposure to labs. But if you don't have that, then don't be shy to arrange lab visits or short lab placements, however you can, to connect to researchers and to ask lots of questions and really not be shy and not be embarrassed by ignorance. I think that's sometimes very difficult to reconcile for clinicians because we're obviously in a very professional role at the time when we go into research, but we're really going into a training post where we're starting at a very low base. As I'd alluded to earlier, I think having a supportive clinical home is very important, and having clinical colleagues who are also invested in your question will be very important long term. So you can't only be doing it for yourself to satisfy your own curiosity and your own observations. Doing your research on behalf of your clinical unit really should be the vision. Mentors are extremely important, and ideally they should be independent of what clinical research training you're doing. So it should be someone who's really outside. And role models, of course, but having acknowledged at the start, they're few and far between in the UK. And I was pleased to see that most of you are either doing a PhD at the moment or are interested in doing a PhD in the future. So hopefully this is helpful. So just remember that it's a really unique opportunity to do something different. So even if you don't end up on the clinical academic path, and the trap that clinical research fellows sometimes fall into is that they'll be tasked with doing stuff during their protected research time that they really could do in the context of their clinical training as well. So things like writing protocols and collecting samples and curating data sets. That's fine, and that may well feed into your research question and research methodology. But if you're in a lab, you really are there to get your science training. So be very protective of that, and don't be put off by labs that are working on fundamental science, because they can teach you something that is very, very different and really open your mind. And I think especially for those who are already thinking that they want a long-term career in science, having a PhD that's that doesn't give you lots of research skills in the lab, wet lab or dry lab or both, is really critical. Otherwise, you'll get to the end of your PhD and you won't feel comfortable enough to carry on. Of course, this does mean getting out of your comfort zone, which that sort of relates to this last point that I've written, which is to leave your ego at the door. I think it's important to embrace the fact that you're entering what is essentially a new field. And I think sometimes as a prop, clinicians use their clinical knowledge to sustain themselves in the lab. So they're the person that offers this sort of the clinical context. And while that can be very enriching for the lab that you are in, actually you are there to learn from them mostly, rather than the other way around. So, I would really let go and enjoy the learning. It is a training opportunity. You don't have to get a paper out of your PhD. It's nice if you do. But the missed opportunity will be not really focusing on your training. And part of that is also to read and go to talks that are both in and outside your field as well, and always talk to people about your work and their work. So how might this trajectory look like? I'm now sort of bearing in mind that there is a different structure these days, and where the funding sources. Well, in the pre-doctoral space, so this is for core and foundation trainees. There are lots of research bursaries. Cancer Research UK is great at these. So if you do have some protected time, and you need a small amount of funding to generate preliminary findings, or really just to dip your toes in research, then these are excellent. There are plenty of clinical research fellowships around, and obviously most medical oncology trainees will go down this path. And the only thing I would say is that doing a PhD is not the only way to experience research. And I think doing shorter higher degrees as well, if you think that you're not going to be on the academic path, can be just as valuable. We've had 2 or 3 fellows who've gone down the MD route, but actually they had really hardcore lab-based projects nevertheless, and they got almost as much out of it, but without getting a PhD. So I think also getting out of that mindset that it's really necessary to do a PhD as a sort of a career building exercise, and that decision about whether you will do a PhD or an MD will be much better informed if you spend a little bit of time in a lab, or at least talking to researchers. And then I think increasingly lots of people will go for a clinical lectureship post, which is facilitated by the NIHR. And I think the attrition really happens mostly between this and then the clinician scientist path to independence. And there are a number of fellowships in this space, and I think several factors have been identified and where this attrition comes from. I think Some of it is that, the part of the clinical lectureship is really to find out whether you want to carry on or not, and therefore legitimately some people decide that they don't. I think for others, the lack of sort of clear trajectory, something that I mentioned at the beginning, is very off-putting, and that sort of insecurity. And there is truth to that, certainly, and much of the subsequent path is sort of organic. It's what you make of it. But I'll come back to the funding in a moment. And then, there are intermediate fellowships or advanced clinician scientist fellowships. So this is, by this point, you're a senior fellow. And then eventually, just like basic scientists, the idea is that you're underwritten by a research institution or a university, and at least as your personal support is concerned, that's secure subsequently. But this is something that people have to negotiate and think about. But you will spend the rest of your career writing program grants so that you can advance your research, and you can get more people into your lab. But that is also the way that you plan your research. So although this is often highlighted as something that's very painful, I would say that most researchers actually find it a valuable process to go through. So this is what I wanted to highlight. Yes, getting funding is not trivial, but actually there is support for clinician scientists now that is stronger than ever, and creating more clinician scientists is part of the industrial strategy and health research strategy for UK. And that's reflected in these figures in terms of the amount of funding that has been given out. This is another question I get asked a lot about work-life balance for clinical academics, and it is challenging. You have obligations to multiple institutions, and these institutions might have very different agendas and strategies. You're managing different teams, a clinical team and a research team. And really you need to be present for both of them. You have 2 email inboxes, for example, that you need to be aware of on a daily basis. Your external roles will be very different, again, relative to these different hats that you're wearing. And what's interesting, because you end up being relatively rare species in both fields, you do get invited into both groups quite frequently, clinical and scientific, because you'll often be the person that provides that sort of unique perspective. You spend a lot of time writing grants and papers, you spend time educating people, clinicians and scientists. There's quite a lot of travel involved. That's the nature of science, but I suspect that is going to change now that we've switched to so many meetings being online. I suspect there will be a lot less travel in the future. There is networking, which is very intuitive in the context of clinical research setting, because you're networking with colleagues that are involved in the same clinical trials as you, and you go to a set number of oncology conferences. But networking in science is a lot more organic, especially if you're at a sort of cross-disciplinary boundary and you're not working on just one specific question in basic science. The clinical work, so for example, my funding, my personal funding is entirely from Cancer Research UK, and that allows me to do 20% clinical work every week, so that's 1 day. And that does then effectively add 1 day to your working week. So why still do it, or taking all this into account, and how to do it? Well, I think the thing is to see a common thread throughout both your clinical and research work, and strive to see how it all fits together. And that also means really integrating your clinical and your research team, and making your research really be as output of your clinical unit, not just your own output. But there's just no denying that you're going to be busy. So recently I was asked to put something together for a report for a funder in terms of the things that I'm involved with. So, there's patient advocacy and editorial boards and peer review, public engagement, scientific committees, lots of stuff with ESMO. I'm involved with clinical guidance. But throughout all this engagement, what the brief that I'm given is to bring my own perspective to these activities. So, for example, if I'm participating in putting together clinical guidance for management of a particular cancer, I come to it from the point of view of translational research and the sort of work that we do in the lab. So, again, having a common thread through all these activities is important. So I just wanted to finish by saying that we have open positions in the lab, both doctoral and postdoctoral. So please do contact me if you're interested. And I talked a lot longer than I had planned, but hopefully we have a bit of time for questions. I'll stop sharing my screen.
Thank you, Samra. That was an excellent, wonderful talk, and it was really nice to hear your sort of personal journey, if you will, through your training. I think we've got the chat function open for questions, so if anyone wants to comment or add any questions, please do so on there. But I would quite like to just kick us off, if that's all right, just by asking, you mentioned sort of in the talk how it's so important to find these positive role models and mentors to help you through the process. I just wondered what your thoughts were on what it takes to be a great mentor, given that some of our attendees may be postdocs at this time.
So identifying a good mentor, is that the question?
Well, what you feel are the traits that make someone a great mentor, and how we can aspire to be great mentors for.
Oh, right. Yeah, of course. So it's very important to sort of separate out mentorship from sponsorship. The latter's very important. And I think we all act as sponsors. It doesn't matter what stage of clinical training you are. If you're dealing with, as a registrar, if you're dealing with SHOs and house officers, and as a consultant with your team, you're giving people sponsorship as they apply for a position, or they're interested in getting involved in another field is very important. But mentorship requires a great degree of objectivity, and that's why I talked about the importance of choosing a mentor that's not directly involved in your field. And I think that's tricky because people are often driven to people that have done something that's very similar to the field that they're interested in. But invariably, their views will be very influenced by their own experience of the field. And while that in itself, that shared experience could be valuable, that's not quite what a mentor is there for. A mentor is a little bit like a life coach, if you like. It's someone who's able to step back and see the bigger picture concerning what you've done to date, and what you plan to do, and probing you perhaps with the less comfortable questions. They're not there to congratulate you and pat you on the back and say, oh, that's all great, you just keep up the good work. To ask questions such as, will this really work? How will it work? Is this what you really want? How does the engagement, how does accepting this role and that role really help you in the long term? So they have your best interest at heart, but they're not scared to raise the difficult questions. And that's why their independence is really important. And it doesn't have to be even someone who is, if this is relating to a clinician scientist career, it doesn't have to be a clinician scientist. It can be, an NHS consultant that you've worked with. So, that those would be my sort of reflections on it. So there's a question. from Alison, what advice do you have on creating a path to independence away from your PhD area? So I think this is where we really should borrow from basic scientists. So the tradition in basic science is that you don't carry on in the same area following your PhD, because PhD is a training program and gives you the sort of fundamental skills to then continue to ask important questions in science. And while they will, they might broadly stay in the same field. So let's say if you're in biology, you'll stay in biology. If you're in structural chemistry, you'll stay in structural chemistry. Going to another institution, choosing another disease model, and for us, I guess, you know, choosing potentially another cancer type or another way to approach a problem is very important, and that's what gives you the authenticity as well. I think replicating something that your supervisor has done is not going to sit comfortably with you, and it won't give you the confidence to think that you can last for the long term. Building on your previous success doesn't mean necessarily continuing to work in the same subject. You will bring a very fresh perspective to another area with what you've done in your PhD. So I really wouldn't fear it. And again, you know, just remember that when we compete for funding and when we publish papers, most of the time we're competing with basic scientists, not with clinicians. And they will have always strived to have as diverse a research career as possible. And that does mean moving institutions, moving laboratories, and you know, being courageous enough to move away from, you know, yet again move away from your comfort zone, but this time, you know, with excellent training behind you. So I think, you know, there is a great advantage, and I certainly felt that, having done, you know, PhD in the context of melanoma, cell signaling, and taking those skills with me, but then really developing in the direction of cancer genomics and building a research area around kidney cancer instead. And you know, I've come full circle now, because, you know, melanoma has remained an interest of mine throughout this time. And you know, we're now in the lab, we're going back to creating projects in melanoma as well. So you might not be letting go completely, but for the sake of independence and, as I said, authenticity as well, it's very important to go and do something that's you know, a separate path.
Thanks, Samra. And I just wondered as well, sort of, you mentioned in your talk about how you found your home really at the Marsden and you skipped that clinical lectureship stage. I mean, how challenging was that to sort of skip that stage of training, and what did you learn from having to miss that out?
Yeah, so I think I was fortunate in my PhD that so as I said, I started off very much working in cell signaling and working on a mouse model, a preclinical model of KIT mutant melanoma. And then in the latter part of my PhD, inspired by NGS, we started probing what rare melanomas look like from a genomic perspective. And there was really no cancer genomics expertise in my lab at all. I mean, it was a, you know, fundamental research lab. So I had to do quite a bit on my own around that time, and sort of build relationships externally, both with, you know, other teams at ICR and also groups internationally who were doing this, to bring about that change for our program. So I think that gave me some grounding in thinking and acting independently. As I said, ideally, I would have done a CL, but I wanted to go to another institution. So I spent quite a long time on my developing my research proposal for the clinician scientist fellowship. I guess if I hadn't been successful in obtaining that, it would have been too late to have gone back to the clinical lectureship. So it was a risk, certainly. But you know, I think by that point I was fairly clear in where I wanted to go with my research. But I think CLs are great if you're not. But bear in mind that sometimes you may be ready for a CSF after your PhD. It depends on what kind of training you've had, what kind of journey you've had.
Brilliant. Thank you so much, Samra. I think we'll have to leave it there because I think the breakout rooms are due to change, but we've all really enjoyed your talk. So thank you so much for giving up your time on a Sunday.
Pleasure. All right. Good luck, everyone.
So my
Bye-bye.
understanding is that Johnny will just now change the breakout rooms, so everyone should be reallocated shortly. So it might be a good opportunity to just grab yourself a hot drink to settle in for the next talk.
Hi guys, as we all start to